Starting with chapter 83, which is pregnancy and lactation. Keep in mind that pregnancy involves all these events: the transport of the sperm and ova, then fertilization, blastogenesis, and then implantation. As far as the maturation and the fertilization of the ovum is concerned, you should know that primary oocyte is when first meiotic division occurs at the time of ovulation. Secondary oocyte is with the second meiotic division. Once a sperm has entered the ovum, the oocyte divides again to form the mature ovum, which now is called a female pronucleus. And it has in its nucleus 23 chromosomes, and each is a single chromosome. Fertilization is the fusion between male and female haploid gametes in the ampullary region of the fallopian tube to produce a diploid zygote. So remember where it occurs in the uterine tube ampulla, and around 100 sperms are needed for it. And when does it occur? After the ovulation. Because the egg, it remains viable 12 to 24 hours post ovulation, while the sperm that stays for up to 72 hours, and then it takes 3 to 4 days journey to the uterus. For fertilization, sperm must penetrate several layers, and acrosomal reaction that allows the sperm penetration. First sperm reaching egg binds to the sperm binding receptors on the oocyte membrane and enters, while the cortical reaction that prevents polyspermy. So here are the stages of fertilization. Penetration of corona radiata and zona pellucida, and then the fusion of oocyte and sperm cell membranes. When one sperm reaches the membrane of the ovum, fusion to the ovum membrane is mediated by fertilin. The fusion provides the signal that initiates the development. In addition, the fusion sets off a reduction in the membrane potential of the ovum, which prevents polyspermy. The changes which take place in oocyte, they include the completion of second meiotic division and the formation of female pronucleus. Next is fusion take place with the male pronucleus to complete the fertilization. 23 unpaired chromosomes of the male pronucleus and the 23 unpaired chromosomes of the female pronucleus, they align themselves to reform a complete set of 46 chromosomes, means 23 pairs in the fertilized ovum. Uh, Now, the fertilization that results in the formation of zygote with the diploid chromosomes after genetic recombination, and then there is initiation of cleavage. Early pregnancy can be confirmed by trophoblastic factor after 24 to 48 hours, while a human chorionic gonadotrophin that can be detected in blood or urine after 6 to 12 days. Let's now look at the transport of the fertilized ovum in the fallopian tube, which is also called a magic tube. The mucosal layer of the uterine tube, it contains ciliated columnar epithelial cells whose function is of course moving, which convey the eggs in the uterus direction and meet with the sperm in the ampulla due to the muscular contractions. Zygote is formed after the egg has been fertilized by the sperm and the chromosomes they are merged from both the gametes. And there is production of embryonic cells at 4 days after fertilization, which lasts for only 4 days in total. On the fifth day, it becomes a blastocyst. Let me remind you that after fertilization occurred, zygote is transported to the uterus due to ciliated columnar epithelial cells by opposite movement due to the contraction of circular and longitudinal smooth muscles. But what is blastocyst? You see, it's a thin walled hollow structure which contains a cluster of cells from which the embryo arises. Now, the developing zygote that implants in the excretory endometrium, when the dividing zygote moves from distal fallopian tube to uterine cavity over a period of 3 to 4 days. Implantation of the blastocyst into the endometrium that happens at around 7 days after the fertilization with about 100 cells. The functions uterus performs after implantation, they are the protection of the embryo or the fetus, then it's nutritional support, and also the ejection of that fetus at birth. Well, once the implantation has taken place, the trophoblast cells and the other adjacent cells they proliferate rapidly, forming placenta and various membranes of pregnancy. Progesterone is generally the hormone that maintains the pregnancy by ensuring a quiescent uterus with no contractions. HCG, which is created by the developing placenta, it prevents the corpus luteum from degenerating and stimulates it to continue to produce progesterone. And at week 7, placenta takes over the progesterone production and corpus luteum degenerates. Now, HCG behaves much like FSH and LH with the one crucial exception that it is not inhibited by the rising levels of progesterone. Thus, HCG prevents the deterioration of corpus luteum at the end of fourth week and enables the pregnancy to continue beyond the end of normal menstrual cycle. Well, HCG on its own can stimulate the testosterone production but not sperm production. On its own, it can also cause an increase in the testicular size and sensitivity as they start to produce their own natural testosterone. And for sperm production, if fertility is required, the HCG in combination with the FSH medication that is required. Hmm. Nutrition. Uh, the early nutrition of the embryo that is achieved from the decidua alone till 1 week after the implantation. The embryo continues to obtain its nutrition in this way for up to 8 weeks, although the placenta also begins to provide nutrition after about the 16th day beyond the fertilization. trophoblastic period of the nutrition, it gradually gives way to the placental nutrition. As the trophoblast cells, they invade the decidua, digesting and imbibing it. The stored nutrients in the decidua, they are used by the embryo. And during first week after implantation, this is only means by which embryo can obtain nutrients for up to 8 weeks. Although the placenta, it also begins to provide nutrition after 16th day beyond fertilization. While we have it, let's look at the anatomy and the function of placenta also. Placenta is a fleshy structure that develops mostly from fetal chorionic tissue and maternal decidua during pregnancy. It lies implanted on the uterine wall, and it is connected with fetus through umbilical cord in the amniotic cavity, and it maintains pregnancy and carries vital fetal functions. So, at full time pregnancy, if we look at the anatomy through naked eye, placenta is a disc like spongy, fleshy structure which is thick at center but thin at the edges, and it weighs 500 grams. Uh, it measures from 15 to 20 centimeters in diameter and 2.5 centimeter at the center. Its volume is around 500 milliliters. Now it has two surfaces, the fetal surface and the maternal surface. The fetal surface is covered with the whitish, smooth, and glistening amniotic membrane and the umbilical cord that is attached at or near the center. And branches of the umbilical vessels, they are visible on this surface radiating from the umbilical cord. Now, this amniotic membrane, it can be peeled off from the underlying chorionic plate except at the umbilical cord. If we look at the maternal surface, this surface looks dull red and it shows 15 to 20 lobes, or you can say the cotyledons, the maternal cotyledons, which are separated by sudden sulci. Now, this surface is covered with the thin grayish layer of the decidua compact layer and spongy layer that comes away with basal plate at the time of separation. The margin of the placenta is formed by the fusion of basal and chorionic plates, and it is continuous with the two membranes, chorion and amnion. Placenta is attached to upper part of the posterior or anterior wall of the uterine cavity near the fundus, and it normally separates after birth of baby through spongy layer of decidua. You should also know about placental permeability in early pregnancy, where the total diffusion conductance is minuscule because the placental membrane is still thick as it is not fully developed, so there is low permeability. And further, surface area is small because placenta has not grown significantly. Placental permeability in later pregnancy increases tremendously because of the thinning of the membrane diffusion layers, and the surface area, it expands many times. Okay, now we move on to the placental functions, which include the transport function, the respiratory excretory function, and also the nutritive function. The placental membranes, they actively control the transfer of a wide range of substances by five major mechanisms, including simple diffusion, facilitated transport, active transport, and pinocytosis. And then there is a bulk flow of water and some solutes, which results from hydrostatic and osmotic pressures. Let's now look at the diffusion of the oxygen through the placental membrane. You see, same principles for oxygen diffusion through the pulmonary membrane, they are applicable. Dissolved oxygen, it passes from large maternal sinuses to fetal blood by simple diffusion, which is driven by oxygen pressure gradient. And near the end of the pregnancy, this mean gradient becomes equal to 20 millimeter of mercury. Although the fetal blood leaving the placenta, it has a partial pressure of oxygen of only 30 millimeter of mercury. Fetus is capable of receiving more than adequate oxygen through the placental membrane by means of the Bohr effect and also fetal hemoglobin, because the hemoglobin concentration of fetal blood, that is about 50% greater than that of the mother. Now, fetal hemoglobin, it is synthesized in fetus before birth, and oxygen dissociation curve for fetal hemoglobin, that is shifted to the left of the maternal hemoglobin. So, at low partial pressure levels in the fetal blood, the fetal hemoglobin can carry 20 to 50% more oxygen than can maternal hemoglobin. Bohr effect explains that hemoglobin can carry more oxygen at a low partial pressure of carbon dioxide than it can at high partial pressure of carbon dioxide. Uh, the fetal blood entering the placenta, it carries large amounts of the carbon dioxide, but much of this carbon dioxide, it diffuses from the fetal to the maternal blood, and loss of carbon dioxide makes the fetal blood more alkaline and maternal blood more acidic. And then there is a double Bohr effect. You see, the Bohr shift, it operates in one direction in maternal blood and in other direction in the fetal blood. These changes, they cause the fetal blood's capacity to combine with the oxygen to increase and that of the maternal blood to decrease. And it forces more oxygen from the maternal blood while enhancing the oxygen uptake by the fetal blood. The diffusing capacity of oxygen at term is about 1.2 milliliter of oxygen per minute uh, per millimeter of mercury oxygen pressure difference across the membrane, and it compares favorable with that of the lungs of newborn baby. Now, for the dissociation of carbon dioxide through the placental membrane, you see partial pressure of carbon dioxide in fetal blood, that is 2 to 3 millimeter mercury higher than that in the maternal blood. Now, this small pressure gradient, that is sufficient to allow adequate diffusion. So, extreme solubility of the carbon dioxide in the placental membrane, it allows the carbon dioxide to diffuse about 20 times as rapidly as oxygen. Excretion of the waste products through the placental membrane, it occurs in the same manner that carbon dioxide diffuses from the fetal to the maternal blood. Mm, Other excretory products, they also diffuse through placental membrane into the maternal blood and then excreted along the excretory products of mother. Uh, Most fetal excretory products, they are non-protein nitrogen such as urea, uric acid, and creatinine. The urea level in fetal blood, that is only slightly greater than that in the maternal blood. Creatinine has a fetal blood concentration considerably higher than that in the mother. Uh, since urea diffuses, um, this diffuses through the placental membrane with great ease, but the creatinine, it does not diffuse easily. Now, the excretion from the fetus that depends on the diffusion gradients across the placental membrane and its permeability. And there is continual diffusion of these substances from fetal blood's high concentration to the maternal blood's lower concentration. Let's now look at the diffusion of foodstuffs through the placental membrane. The metabolic substrate diffuse in the same manner as oxygen does. For example, the glucose that is transported by carrier molecules in membrane's trophoblastic cells. In later stages of pregnancy, the trophoblast cells lining the placental villi, they provide for facilitated diffusion of glucose through the placental membrane. These diffuse from maternal to fetal blood, but more slowly than the glucose, the other the other nutrients. So, glucose is used more easily by the fetus for the nutrition, while the ketone bodies and the ions like potassium, sodium, chloride, they diffuse with relative ease from maternal to fetal blood. And then there are some other functions also which are performed by placenta, and those include the enzymatic functions, storage functions, barrier function, and immunologic function, and also the endocrine functions, which are very important. You see, the placenta, it produces hormones that are vital to the survival of the fetus, and these include the protein hormones like HCG, human placental lactogen, and they also include the steroid hormones such as estrogen in all the forms and the progesterone.